Hey class, and welcome to your next part on evolution and adaptation. So we started talking all about what evolution is, what adaptation is. We talked about Darwin, natural selection, and in class you learned about the different forms of natural selection. So now we're going to go into a little bit of evidence for evolution. We will spend a little more time on this, but I just want to get you started here. So our first evidence for evolution is that scientists observed natural selection in action. We can actually see it happening, and I'm going to give you a few examples. So Rosemary and Peter Grant have worked in the Galapagos Islands for over 20 years. Now remember, the Galapagos Islands is where Darwin went and he studied all those finches. And today, we are still studying exactly what he studied, and we're able to see natural selection happening. So remember, natural selection is just the survival of the fittest, fittest meaning that it can pass on its traits. So we want to be able to pass on our traits to our offspring. So they've been studying in the Galapagos Islands for over 20 years, and they've noticed that in wet years, small seeds are more abundant and birds with small beaks are favored. But yet in dry years, large, strong beaks are favored because large seeds remain. So in one year, we might have these small birds and with small beaks because it's wet, and that's going to be what is favored. But the dry years is going to be exactly the opposite because that is what is left over. So continuing on with scientists observe natural selection in action, think of the development of pesticide resistance in insects. And we're going to go over this with insects. We're also going to go over it with bacteria. So the development of pesticide resistance in insects. What happens is initially the farmer or whoever is growing crops will use a pesticide. And that initial use of pesticides favor those few insects with the gene for pesticide resistance. Now, while I'm going through this, think about what type of natural selection this is. And go ahead and just write it down somewhere, and we'll talk about this a little later. So we have that initial use, and it's going to kill off quite a few bugs or insects because they do not have that pesticide resistance gene. But then they're going to continue using it and continue using it, and they're going to see that more and more bugs are coming by, and they might think this is the product. The product's not working anymore. But in actuality, the product is still working, but more and more insects have this gene for pesticide resistance because they're the ones able to reproduce and produce offspring which are then resistant to that pesticide. So those without that gene are going to die off, and this is due to their environment. This environment has this pesticide, which is killing off any insect that does not have this pesticide resistance gene. And what's going to happen is that proportion of resistant insects is going to increase over time. More and more and more insects are going to be around there because that pesticide is not killing everything that it needs to kill. Now, I said to write down what you thought this was, and if you said that it was directive, you would have been correct because we are favoring for one gene. This one gene of this pesticide resistance is what we are favoring for and what we want. Now I'm going to go through the same thing. It's not on a slide, but it is for methicillin resistant bacteria. And we generally call this MRSA, and you're going to hear about it. A lot of times you'll hear about it in gyms, but in hospitals too. But in actuality, it could be almost anywhere. This is due to our overuse of antibiotics. Now, antibiotics are a very, very good thing. These antibiotics are going to help you when you have strep throat or when you get a staph infection. Any kind of infection that is bacterial in nature, these antibiotics are going to be very, very good. But the same thing is going to happen that happens with the pesticide resistance. And what's going to happen is initially, the first couple years that we're using it, it's going to kill off 99.9% of those bacteria. And they're not going to be able to reproduce and pass on their traits. Now, this is where it becomes a huge problem in hospitals. Think about your hospital rooms, and think about your ORs and how clean they need to be. So we're using all kinds of products to kill off these bacteria because we don't want to get infections when we have surgery or we're in the hospital because we have an open wound or something. And so we're constantly trying to get rid of these bacteria that are in these hospitals. But that 0.1% that has this gene for resistance is then going to start to multiply. And when they start to multiply, our drugs are no longer useful for them. So they become what we call superbugs. And these superbugs seem to be very hard to be able to kill off. So a lot of times we find these in hospitals because we're trying to keep them so clean with the alcohol, with antibiotics, anything that's going to kill them we're going to try and use, and this becomes a major problem. Okay, so you have quite a few examples on natural selection in action, but I want you to come up with your own. Write about a paragraph about it, how it got started, what that environment is like, and we're going to turn this in on Monday. Okay, so our second form of evidence for evolution that should be fossil record, not fossil fuels. So make sure you go ahead and write down fossil record. Alright, so the fossil record shows that organisms have evolved in historical sequence. And we're going to look at different forms of how we have evolved. But I'm sure you've heard of analogous and homologous structures. We're going to study those. But the fossil record just shows this historical sequence. And our oldest known fossil is that of a prokaryote or bacteria. Very, very, very simple. And we can see based on the fossil record that we start adding more and more and more to these eukaryotic cells, or these prokaryotic cells, to make eukaryotic cells. And billions of years later, after the first prokaryotic cell, we have the eukaryotic cell. And we know this based on our fossil record. And remember, with fossil record, we generally use carbon-14 dating for the half-life to determine the age. Now, it took quite a few years later to get these multicellular fossils, and they're much newer. If you go to the Field Museum in Chicago, or even the Natural History Museum in New York, both of these have all kinds of fossils, and they show the fossil record. And paleontologists are constantly working on this fossil record to determine where each animal came from, what was its ancestor, what was the environment it was in that its traits changed. And these traits aren't going to change very quickly. As you can see here, the prokaryote to the eukaryote took billions of years, and multicellular animals and plants and organisms came much later. So paleontologists who study fossils are constantly working on this. So lastly, we have, and that should be fossil record. Again, very, very sorry, guys. Many fossils link early extinct species with species living today. So what does that mean? We've seen a series of fossils, and they document the evolution of whales to land mammals. What does that mean? No, whales are not making land mammals. It's the other way around. Over a long period of time, land mammals evolved into whales. And I talked a little bit earlier about these analogous and homologous structures. We will go more into that in depth in class, but just kind of write that down as a note that we need to still talk about this, and this may appear on your quiz. Now, I know I told you in class that your quiz is going to be Tuesday, but it's been changed to Thursday. So go ahead and write that down. We're going to do a little more activities before we have that quiz. Okay, well, have a good rest of the weekend, and we will see you on Monday.